Hi everyone, welcome to my channel Pure Biology, and today's topic is about the shoot apical meristem development, but here the genes involved and it is explained in the molecular way because the genes sometimes involved in the formation of the shoot apical meristem. Okay, so what is that? The mutation of the two genes that are very important. One is the STM gene and another is the WUS gene or washer gene and the shoot meristemless gene. The mutation of it was observed by the scientists that the mutation of these two genes that result in the seedlings with no or reduced SAM means shoot apical meristem. That means mutation of two genes creating no or reduced shoot apical meristem. Okay, and it was observed in the seedlings. Other one gene, that is the CLAVATA, CLAVATA or the CLV, whose mutation result in the enlarged shoot apical meristem. So its mutation, these two genes mutation that showing the no or reduced shoot apical meristem, and CLV gene whose mutation showing that the enlarged shoot apical meristem. Clear? And now the products of the two genes STM and WUS that act as act on the meristematic cells and in the undifferentiated meristematic cells. Obviously, these act on the region of the meristem, or you can say the meristematic region cells, and which cell that are undifferentiated cells. Clear? WUS gene that encodes one transcription factor, that is the homeodomain transcription factor, and the gene is expressed in the small region immediately under the stem cells and the known as the organizing center. So where this WUS gene expression is seen? This is seen where? This is seen in the under the stem cells known as the organizing center. Where it is seen? Under the stem cells. Under the stem cells. Where? Under the stem cells, which is known, specifically this region is known as the organizing center. And this is very important. Why? Because this is critical for maintenance the shoot apical meristem, because organizing center actually maintains the shoot apical meristem cells division. And here this is caused by the adjacent cells to acquire the stem cell fate and remain indeterminate. So what is saying that this organizing center is the place where the expression of WUS gene this is seen, and this is critical for maintenance of SAM. By how this maintenance is controlled? By causing the adjacent cell, that means the organizing center. Surrounding this organizing center, there are many cells, or you can say these are the adjacent cells. These acquire the stem cell fate. Okay, these acquire the stem cell fate and remain indeterminate. Okay, they behave like stem cell fate, and that's why they remain indeterminate. Okay, so in that way, this is controlled. Now, WUS expression that define the organizing center of shoot apical meristem, and which is a group of cells just below the stem cells. So this WUS gene, what is the role of this? This expression define the organizing center. That means if this WUS gene expressed, that means which cells form the organizing center, that will be determined. Clear? And this STM gene, so I think you can understand it. Okay, I just briefly again explain that this WUS cell, this is expressed where? This is expressed in the organizing center. Okay, this is expressed organizing center, and its role is what? It's generally, what is the role of this organizing center? This actually determine, or it helps to determine the shoot apical meristematic cells. Clear? Shoot apical meristematic cells. Now, this STM gene's role is what? This STM, that like WUS cell that encodes a homeodomain transcription factor, likewise STM gene that encodes another transcription factor and acts independently of WUS to prevent the differentiation of the stem cells. So just opposite to the WUS, because WUS actually help to determine the formation of the organizing center, but STM gene's expression that helps to prevent the differentiation of the stem cells, or it prevents the organizing center to differentiate the stem cells. Clear? But this is absent where? This is absent in the leaf primordial. Okay, the expression of this gene, STM gene, is absent in the leaf primordial. Clear? So what is the role of WUS gene and the STM gene? It is clear now. Okay. Now another gene, that is the CLAVATA, CLAVATA. It has the three subfamily or what I am saying, that subfamily you can say, or the three types of gene is seen there. The CLV1, CLV2 and CLV3. These genes generally control the size of the shoot apical meristem and also the floral meristem, because if the mutation of this CLVATA gene is present in any plant, that means the plant's size become very enlarged. Because SAM or shoot apical meristem's size also be enlarged, that's why the plant size also be increased. So role of this CLAVATA is what? It control the size of the shoot apical meristem and the floral meristem. Now CLV1, this encodes leucine rich repeat receptor kinase. Short form is that LRRK, leucine rich repeat receptor kinase. This is another protein obviously. In absence of ligand, because ligand always bind with the receptor. So if the ligand is absent, the receptor is inactive. But when the ligand is present, then the receptor becomes active. Clear? And in that situation, this protein can bind the kinase, obviously, that bind with the receptor. Okay. Or the factor, you can say, the transcription factor that can bind with the receptors. The ligand for CLV1 is a small protein and encoded by CLV3. So the protein that is bound with the CLV1, it is bound with the CLV1 with the help of CLV3. Clear? And the CLV2 that encodes a receptor similar to CLV1, but this has no domain. Like same thing is happening in the CLV2. CLV1 like receptor is there also in the CLV2 for binding with the ligand, and but there is the domain absent. Clear? So what is saying here, that CLV1, it encodes one protein. This is called the leucine rich repeat receptor kinase. When the ligand is absent, this kinase remain inactive condition, and if the ligand is there, ligand is bound with this kinase, that means kinase becomes activated, and now transcription factors that is controlled by this receptor. Okay, so the size of the organizing center is regulated through a feedback loop involving CLV and WUS. Obviously here WUS gene has an important role, or important role is played by this gene for controlling the size of the shoot apical meristem. Clear? CLV that negatively regulate WUS gene and WUS that stimulates the activity of CLV3 encoding a secreted polypeptide. This is another factor. So its control is dependent on the WUS because it stimulates the activity of the CLV3. So the protein CLV3 binds with the CLV1 and CLV2 receptor and induces a signaling pathway that inhibits the WUS and limits the size. So how the limit is controlled by this CLV? That there is a protein, that protein when binds with the receptor then induces a signaling pathway, and due to this arising of signaling pathway that inhibits the activity of the WUS, and that's why the size of the organizing center is also limited. Clear? Now CLV3 expresses where the layer 1 and layer 2, that means the cells present in layer 1 and layer 2, divisions controlled by this CLV3 gene. But CLV1 that is present, or that expression is seen in the layer 3 containing cells region or the central zone. So this is the briefly explanation of the shoot apical meristem development or molecular aspects of shoot apical meristem. If this is totally understood by you, then please give a like. That help me to motivate the to continue, you can say, to motivate me to continue the channel. Okay, and if this is helpful to you, so please you subscribe this channel and share this with your friends. Okay, so thank you.